Welcome to Ask the Expert from the Sugar Science. It's a new style of scientific communication. It's kind of more like a town hall than a lecture. It's short to accommodate the time-sensitive world of scientists, and we believe that collaboration accelerates scientific research, and especially across disciplines. Today, for our guest, I am very excited to have Zachary Freyberg, MD, PhD. He's currently at the University of Pittsburgh. He's a physician scientist with a basic science training in cell biology, imaging, genetics, neuroendocrinology, as well as clinical training in psychiatry. So he has really a great reach across all these disciplines. And as an independent investigator, Dr. Freyberg has combined his basic science and clinical expertise to develop his current research focus on peripheral dopaminergic mechanisms of metabolic regulation and their roles in diabetes and antipsychotic drug-induced metabolic disturbances. He received a BS at Yale University in the MD&B department, and then he received his MD and PhDs at Albert Einstein in following. So during the PhD, he investigated the roles of lipid signaling and controlling hormone secretion in the laboratory of Dr. Dennis Shields. And after completing his clinical training in psychiatry at Cornell, he trained as a postdoctoral fellow at Columbia in the labs of Jonathan Javich and Dave Sulzer, focusing on mechanisms of dopamine neurotransmission. So this kind of led him to his current studies, examining the roles of dopamine signaling outside the brain in organs like the pancreas, and how medications like antipsychotic drugs alter the signaling to cause insulin resistance and diabetes. So welcome, Zach. Thank you so much for joining us. I can't wait to talk about your new paper, which is New Roles for Dopamine as a Pancreatic Modulator of Insulin and Glucagon Secretion. And thanks so much for joining us.
Well, first of all, thank you so much for inviting me. I'm humbled and grateful to have this opportunity for us to chat and to speak with the larger community. This is obviously very exciting, and I hope that we can discuss some interesting ideas and maybe find a new horizon, as it were.
I love that idea.
Cool. So should I, I have a few slides.
Yeah, let's dive into those. Yeah, let's take a look at what you got.
Okay, perfect. So I guess I'll start. Can you see? Or should I share screens?
Yeah, share your screen.
Okay, wonderful. Let me start that then. Okay.
There we are.
Cool. All right. So I'll go into presentation mode. So very briefly, right, the slides that we have here relate to today's topic, which is new roles for dopamine as a pancreatic modulator of insulin and glucagon hormone secretion. And, while dopamine has been studied for at least 60 years, primarily in the brain as a neurotransmitter, it turns out that we actually have decades of data suggesting that it does lots of stuff outside of the brain. And in fact, some of the first studies to give evidence of dopamine's role in metabolism come from Parkinson's disease. It turns out that when patients with Parkinson's disease were being tested with L-Dopa, which is one of the mainstays of treating the motor symptoms of Parkinson's, they actually started developing metabolic symptoms. They started developing hyperglycemia, elevated blood sugar, which was totally unexpected. And that led to why. So it turns out that dopamine, which is the product of L-Dopa, inhibits insulin release in pancreatic beta cells and islets. And even more interestingly, these same beta cells actually express the same dopamine receptors expressed in neurons, and in particular, dopamine D2 receptors. So dopamine D2 receptors are inhibitory in the brain. They inhibit the release of neurotransmitters, including dopamine. So what my colleagues and I found was that this same receptor is an inhibitory receptor for insulin secretion, and in particular, glucose-stimulated insulin secretion. But in vivo on a systemic level, it still hasn't been clear or entirely obvious whether metabolic effects that are mediated by dopamine are primarily a result of dopamine's actions in the brain that cascade down to other organs, whether dopamine acts on these same dopamine receptors primarily in the periphery outside of the brain in order to cause metabolic symptoms, or probably, most likely because, life is messy and we deal in shades of gray, it's probably some combination of all of the above, where one system is affected and it feeds back on the other and vice versa, because nothing functions in a vacuum after all.
Right. And as we know, the brain and the pancreas are connected by the vagal nerve.
That's exactly right. And in fact, more and more evidence suggests that actually might be how Parkinson's disease actually begins, that abnormal protein aggregation happens outside of the brain and tracks up that same vagal nerve to get into the brain. So there's a lot of back and forth. So again, what my colleagues and I then did was, well, if some of these components of the dopamine machinery, which have been well characterized in the brain, are important functionally, more on a more basic level, we stepped back and looked first in beta cells, but then also in other important pancreatic islet cell types like alpha cells, the cells that produce glucagon, the hormone that raises blood sugar, to see whether they express dopamine receptors as well as other components of the dopamine biosynthetic machinery. And it turns out that both alpha cells as well as beta cells express some combination of all 5 dopamine receptors. And they also express the biosynthetic enzymes, including tyrosine hydroxylase, the enzyme that converts tyrosine to L-DOPA, and dopamine decarboxylase, which takes the L-DOPA and turns it into dopamine, as well as the transporters, proteins, which actually shuttle in L-DOPA that circulates in the bloodstream into pancreatic islet cells to convert them into dopamine. Those are called LATs, large neutral amino acid transporters. And so as you can see here, they're in deep red, suggesting that both alpha cells and beta cells express bucket loads. What's particularly important about this is that when one eats, levels of circulating L-DOPA rise 50-fold, so that you can then couple peripheral dopamine synthesis to meals and meal size. So the next question that we sort of investigated, first in beta cells and later in alpha cells, was, well, if they express the machinery of dopamine synthesis, do they actually make much? So just taking beta cells from islets or beta cell lines, we just took a look to see how much they secreted, or how much was inside for that matter. And in the absence of any kind of dopamine precursors like L-DOPA, we saw virtually nothing. But when we added L-DOPA, suddenly they started making significant quantities of dopamine, and they could break it down as well, just like neurons, so that it doesn't stick around too long. And we saw, interestingly enough, something similar but different in pancreatic alpha cells. So it turns out that unlike beta cells, in the absence of L-DOPA, alpha cells could make their own L-DOPA independently of any substrate uptake, as well as their own dopamine, and interestingly enough, their own norepinephrine. But when you supplement with L-DOPA, it goes up substantially. It goes up almost 1,000-fold.
Yeah.
Suggesting that alpha cells have the capacity not only to independently make their own L-DOPA and dopamine, but that they can readily take up the precursors. And interestingly enough, even though they express the machinery to convert dopamine to norepinephrine, because these are virtually identical molecules, they preferentially secrete dopamine and not norepinephrine, suggesting that it's dopamine that's the coin of the realm, that's primarily what's being used
I love
to
that
signal.
phrase. That's fantastic.
Oh, cool. So once they make it, what happens next? Well, it turns out that L-DOPA is actually preferentially taken up when beta cells are stimulated. So there's a doubling in L-DOPA uptake during high glucose stimulation, suggesting that, again, this is yet another way of coupling local pancreatic dopamine synthesis to meals when blood sugar rises. Because after all, if this dopamine signaling is inhibitory, then you would want to be able to finely tune the level of inhibition to the degree of stimulation. And this is exactly what we see.
So Zach, at this point, are you hypothesizing that this is like a tuning situation going on here, a fine tuning of the actual, mechanism of
That's exactly
secretion?
right. Yeah, that's exactly what it is. It's a modulator. It's meant to tune in response to levels of food. So for example, right, you don't want to have a one-size-fits-all model of controlling the duration of insulin secretion. So if you have a candy bar versus, a 10-course meal, you don't want the same level of inhibition. So the more you eat, presumably the greater the level of circulating L-DOPA, which gets taken up to form more dopamine and therefore more inhibition.
Yeah.
And so what we find is in both human islets as well as in mouse islets, you see a concentration-dependent inhibition of glucose-stimulated insulin secretion according to the levels of L-DOPA. Interestingly enough, human islets respond in a much more sensitive way to the dopamine compared to mouse islets. And that that's a whole other story, which we recently published, but that has to do with the relative amounts of dopamine receptors to adrenergic receptors, because it turns out the dopamine can signal through both. And you see this here actually when we look at glucagon as opposed to insulin. So what we find is that unlike beta cells, picomolar concentrations, vanishingly small amounts of dopamine are capable of diminishing glucagon secretion. However, after a certain point, when you start passing the micromolar levels, it actually becomes a biphasic response and starts becoming stimulatory. And that has to do with dopamine's ability to actually, at much higher concentrations, to start signaling through receptors other than dopamine receptors, namely adrenergic receptors, because it turns out that alpha cells predominantly express beta adrenergic receptors, which are stimulatory. Beta cells, on the other hand, express alpha adrenergic receptors, which, like dopamine D2 receptors, are inhibitory. And the relative amounts of these adrenergic receptors to the dopamine receptors, coupled with the relative amounts of circulating substrate, the dopamine that's around, determine the type of response that you get.
It's it's so much more complex than you might even imagine at first glance from the textbook, description. So,
yes. I mean, it's still shocking to me that only now are we beginning to move away from beta cells, right, and look at the sociology of the islet, to look at some of these other cell types and the parallels with what you see in the brain with all of the different types of neuronal populations and the diversity of, products that neurons release locally to signal with one another, in many ways is really quite similar to the islet. Yeah. So being able to
it's very orchestral in nature, right?
Yes, that's exactly right.
Can I ask a quick question?
Oh, yes, of course. Hey, Nair.
Hi,
Hi.
that's really nice talk. I never heard about this dopaminergic, let's say, signaling in beta cells or alpha cells. I know a little bit about the vagal innervation and also the beta 2 adrenergic receptors, but I'm like so fascinated by that. So, okay, my question is the signaling, do you know if it's mainly by paracrine signaling, let's say from like alpha cells, since you said that they can also produce dopamine, or it's mainly by the dopaminergic neurons? Or like, what are your thoughts? Like, where is this coming from?
So it's coming primarily locally, it's coming from, so both alpha cells, so where alpha cells can make L-dopa and dopamine, beta cells can't generally make much L-dopa, but they make their own dopamine too. And so what's happening is that you're getting a combination of autocrine and paracrine signaling, which is happening. And that, and you know, at baseline. And then once the circulation starts bringing in L-dopa as a substrate, both of these cell types can readily take it up and convert it into dopamine.
Okay, I see.
So they're signaling, they're communicating to one another. Probably where the paracrine signaling happens most, and this is my guess for now, is under basal conditions in the absence of high glucose stimulation, because at that point, it's the alpha cell, which is freely making L-dopa, that can then get taken up by the beta cell to be converted into dopamine.
Yeah, makes sense. Thank you.
Sure. So that basically leads to, you know, a model, a picture, as it were. So it could very well be. So if we know in both alpha cells and beta cells that dopamine's capability to signal through endogenously expressed dopamine receptors is inhibitory and inhibits insulin and glucagon, then somehow diabetes, whether it's type 1 or type 2, entails dysfunction of these receptors, which leads to both hyperglucagonemia and an elevation in glucagon, as well as an elevation in insulin. And that elevated glucagon leads to hyperglycemia, which potentiates insulin resistance, which then leads to further insulin secretion on top of already the hyperinsulinemia that's happening from, you know, disrupted dopamine signaling to begin with, to create a closed loop where you end up potentiating continued dysfunction.
And that's very toxic to the beta cells, hyperglycemia.
Yes.
And let's just say, just comment for one, I don't know if you're going to get to this after, but you know, when some of these patients go on psychiatric drugs, psychotropic drugs, they encounter metabolic dysfunction, right? So, you know, how do you imagine that you know is impacting the system?
So we actually just published a paper on this, because antipsychotic drugs really can't tell the difference between, you know, dopamine receptors in the brain versus the periphery. What we find in islets is that these same antipsychotic drugs, for example, bind directly to the dopamine receptors.
To all five flavors?
Well, no, they're primarily D2 and D3.
Okay.
There's one that called clozapine, which is actually the worst offender, which also binds D4, although alpha cells and beta cells really don't express much D4. But the single unifying feature of all antipsychotic drugs is their ability to block D2 and D3. And if you take that away and keep their affinities to other targets the same, they lose all their psychiatric properties. So, you know, fundamentally, what we find is that drugs that interfere with this local dopamine signaling lead to dysregulated insulin and glucagon secretion. And in fact, what we found was that the levels of increased glucagon secretion from human islets was in direct proportion to their metabolic liabilities. That's actually how I got into this work. It was as a psychiatrist, because almost all, if not all of these drugs rendered people taking them with dysglycemias, with impaired glycemic control and or insulin resistance. I've seen some patients gain 30 pounds within the first month of beginning these medications. And it's a very hard road to trod. I mean, like, you want to help people who, you know, who are struggling. And at the same time, how do you help people, you know, balance the metabolic liabilities of the drugs with whatever benefits that they can get?
Because now maybe they have, you know, more of a you know, sort of a better metabolic, a better, you know, mental state, but now they're, you know, really at risk for a whole host of another set of cohort of diseases.
Exactly. And in fact, you know, for most people taking these medications, that's the number one reason for why people stop taking them, which then leads to psychiatric decompensation. So, you know,
circle. Also,
yeah.
Isn't it true that the D2 or D2-like receptors in the rat, they decrease intracellular calcium levels? And that, of course, right, has a lot to do with exocytosis.
That's exactly right, as well as cyclic AMP.
Yeah.
Right. I mean, so the pathway that's best understood for dopamine receptors, D1-like receptors, which are stimulatory, and D2-like receptors, which are inhibitory. The reason why they're called stimulatory and inhibitory is because of their effects on cyclic AMP synthesis. And cyclic AMP is, like calcium, one of the main regulators of exocytic vesicular release.
Yeah. Very interesting today. A paper came out. Kevin Herold, you know, had this this paper and others that, you know, is talking about TET2 controlling the responses of beta cells to inflammation in the autoimmune diabetes. And it turns out that, you know, TET2, I think, is also kind of impacted, you know, by like there's an abnormal conversion of alpha-ketoglutarate, right, in the TCA cycle, and that decreases the these alpha-ketoglutarate dependent enzymes, like those that are controlled by the TET family of genes. So it's you know, there's something really, there's so many pieces to this puzzle, right? There's the TCA cycle, there's the receptors at the surface, there's the exocytosis of the insulin and its feedback loop. But I mean, I guess I brought up that paper because, again, they're sort of pointing the smoking gun at the fact that it's actually the beta cell that's dysfunctional that sets the whole autoimmune scenario in motion. So
I'm not convinced of that.
Yeah. And why?
Yeah.
What would you say? Because I mean, this has been the big debate, right? Is it, you know,
Right.
murder or suicide?
Right. Either way, right, the system becomes dysfunctional. And in a sense, you know, it's almost an academic argument, you know, because, you know, one of the things that I think is interesting is that whether it's type 1 diabetes or type 2 diabetes, or even antipsychotic induced, you know, metabolic disturbances, they have something which is unique in the sense that they have both hyperglucagonemia and hyperinsulinemia. If the system were working, you should never, ever have that.
Right.
Right. And yet both are elevated. Hyperinsulinemia by definition should prevent hyperglucagonemia from happening. There's evidence that blocking glucagon signaling blocks the development of these dysglycemias and impaired glucose tolerance in spite of continued problems with insulin. Yeah. And I think that this goes to, you know, a provocative hypothesis that people like Roger Unger, you know, talked about for decades, which is in fact that it's alpha cell dysfunction, which drives the process, and that, the islet being plastic, right? It's the beta cells that are responding to persistent alpha cell dysfunction that are reacting rather than driving the phenomenon.
Yeah, there's a whole set of scientists that are actually really kind of cluing in or honing in on that idea that alpha cells have a real role in this. It comes to mind, a whole group out of Indiana University. But there's also, we haven't really talked that much about this other piece. And that is, Philippe Blanco's group did this, they did an ablation of the pancreatic nerves, and they saw that they could reverse, type 1 in mice, not mice. And then Matthias von Harris' group, his paper that was in Science, interference of pancreatic sympathetic signaling halts the onset of diabetes in mice. So, it's like, I'm just to hypothesize and throw this out here, how might sympathetic innervation impact alpha cell dysfunction? Could it, or could it?
It absolutely could. So one of the things that we found, again, talking about mice for a minute, because,
yeah, it's a little different.
Right. It's a lot different, actually, in terms of sympathetic. And from the sympathetic standpoint, largely, I think, or at least partially, because of the receptors and the receptor availability. And this becomes really important for sympathetic innervation, because what comes out of sympathetic nerves? Norepinephrine.
Yeah.
Right. And what you end up having, and this is what we found actually in our recent paper, was that if the predominant in mice, the predominant GPCR or catecholamine receptors are beta 1 and beta 2 adrenergic receptors. These are stimulatory. So when you have sympathetic innervation, and that norepinephrine is binding beta 1 and beta 2 adrenergic receptors, you're going to be driving glucagon release. So you're generating hyperglucagonemia.
Yeah.
So the more you drive sympathetic, the sympathetic system, the more glucagon you're going to be secreting, and the greater the degree of hyperglycemia, which would then fit.
Yeah, the whole system gets worn down. What has anyone done a full characterization of the the receptors in the alpha cell and their response? I mean, has anybody done a patch clamp of the alpha cells? I'm going to start looking for that.
Yeah. So again, it's a drop in the bucket. Like if you like just go on PubMed and you graph the amount of alpha cell centric papers versus beta cell centric papers, it's like 1 to 40.
Yeah.
People are just beginning to sort of awaken, to the possibility that alpha cells are doing stuff that's relevant.
And the delta cells are hanging out there too. I mean, I think, I think they've been historically hard to replicate also. I mean, when we're talking about islet transplantation, they've got beta cell, locked down. The alpha cell, I think, is fairly, maybe they're getting close to that. I think delta cell has been really hard to figure out and make from stem cells.
Oh, absolutely. Absolutely. Because they're really, if alpha cells are understudied, then the delta cells, like the George Harrison of the islet,
Oh.
Unfortunately, I love George.
I know.
And it makes me sad, but I mean, let's talk in hopefully 2 years about the delta cells, but I agree with you. I mean,
it's a wide open frontier for young postdocs and graduate students, people to start thinking about the fact that it's not all wrapped up. This is really, this is a massive project that still has a lot of information to be gleaned, and it's the metabolic. I mean, it's the nest system of the body's ability to eat, right? So. And I think there's so much work to be done here. It does just sort of disappoint me when I hear like, oh, type 1 diabetes is all wrapped up, we've got the islet implant coming. You already have all this control of the blood sugar via, pumps and CGMs, et cetera. That I would argue that control is limited, and we'll see how long it takes for the islet implantation. But I do think that that these sort of silent partners, I guess, in the sense that they haven't been looked at, need really to come center stage. And I think your work is doing that, Zach. I'm really appreciative of it.
I mean, and there's just the thing about all of this that's been eye-opening for me is I'm coming at this, I've been working on this now for 12 years, but I came at this as a neuroscientist and a cell biologist. And so I had to sort of learn all of this in a very naive sort of way, which I think was good, because what it forced me to do is to see that none of these systems exist in a vacuum, that they're using the same types of signaling motifs, the same types of, especially the same types of conserved receptors that are just coupled slightly differently or to different outputs. But, evolution is smart in the sense that if every organ system, if every cell type had to create these systems to control cell function from scratch, it would just be too energetically expensive. And so the brain is probably the last one to the party. I think that, every organism from bacteria on up have to feed, they have to metabolize nutrients.
Yeah.
And I think that rather than thinking about dopamine and these signaling systems as, neuronal, really, I think the neurons are probably the last of the specialized systems that have basically adopted these pre-existing mechanisms.
What about this idea that with, the Coxsackie insult, that, the viral driver that is really just making things go awry? I wonder if you have any thoughts about whether or not alpha or beta are more, there's been a big focus on the beta cell. It's highly productive, secretory. Once a virus gets in there, everything goes to heck, and and the dysfunction begins there. So, but what about alpha cells? Has anyone looked at what happens? We have the perfect storm here with COVID, but also with Coxsackie. Like, what happens to alpha cells once they're under viral assault?
I personally don't know if anyone's ever looked.
Okay, here's a project.
Yes.
I think there is a review from Zachary that he compares why alpha cells are less susceptible to viral infection compared to beta cells. I think from, 2018 or 2019.
Okay.
It's kind of newish.
Who is it? Who is that now?
Desioeizirek. He is from Brussels, but now he has a lab also in Indiana University. He studies mainly beta cell stress.
Mm-hmm. He's next on the list to call.
Oh, this is fascinating.
Yeah. But I don't know the answer. I forgot.
Well, you can't, can't have every single paper in the world at our fingertips, our mental fingertips, but it's a really, really curious question. And in terms of the market for, let's just sort of talk about the finances. The market for type 1 diabetes is a lot less than the market for type 2 diabetes, right? So some say, okay, well, there's not a lot of incentive for the big companies to get into sort of like drug development for type 1 diabetes, et cetera. But the market for, I don't know what the market for, mental health drugs is, but maybe that's a way to entice some of these larger drug companies in to sort of explore this, how to make a better, psychotropic, so that, and then maybe in doing so, explore some of these paradigms with the alpha cell.
I think that would be wonderful. I mean, I think that the market for psychotropic drugs is complex, particularly for, in the last few years, just because of the lack of success at moving away from the same types of conceptual paradigms that have dominated the field for 60 years. All antipsychotic drugs work through dopamine receptors. People have tried to move away from them precisely because, the original drugs caused Parkinsonism. The newer drugs caused, diabetes. And there's been very little success. And so there's been, many companies have liquidated their CNS divisions or dramatically scaled back. More recently, there was, a recent drug where olanzapine, which is one of the worst offenders, but, in terms of metabolic side effects, but also, probably one of the better antipsychotics, was bundled with another medication, which dampened some of the side effects. I think, so that was sort of a breath of fresh air. What is also interesting, on the other hand, is that there actually is already an FDA approved drug to treat type 2 diabetes, at least, bromocriptine, which is actually a D2 agonist. So whereas antipsychotic drugs cause these metabolic symptoms by blocking D2 signaling, there's a drug that's literally doing the opposite. And perhaps unsurprisingly, it improves insulin sensitivity. So there's been no mechanism, but it's generic. And so the financial incentive is probably lower.
Wow. Yeah. I'm sure it is lower. It is really, I think it is a place where are all the clinical trials on these patients that take these medications, and have there been any repositories, blood repositories, pancreatic repositories, anything of that nature established?
So I'm not familiar with any repositories for patients taking antipsychotics for the pancreatic, at least. I know that, there are several NIH funded brain banks where you have, from subjects who have schizophrenia, who donated their brains, for example, to study the mechanisms of schizophrenia in the brain. But I'm not familiar that other organs were preserved. In terms of clinical studies, we're actually trying to get one off the ground where we're administering low doses of bromocriptine, this agonist, alongside antipsychotic drugs to see if they can blunt or maybe even reverse these antipsychotic induced metabolic symptoms. At least that's a start. Yeah,
maybe a great idea to have these folks, wearing a CGM and seeing what comes out of it. But it might be a good question for Mark Atkinson to ask whether or not NPOD has any sort of subclasses in their collection of pancreata that came from people who took these medications over years.
That would be amazing, actually.
Okay, another email has to be sent this morning. This has really been so interesting, Zach, and thanks, Nara, for joining and asking some great questions. I really can't wait to see what you do at Pitt. University of Pittsburgh has a lot going on, I know, right now, and it's a very exciting, I think, place to be.
It certainly is, and there's a never-ending amount of work.
Yeah.
but just being able to get a foothold and to try and get at this. I mean, there are details of this work that have been around for decades. It's putting it together, I think. I mean, Pietro de Camilli published several papers in the late 80s and early 90s with like Franco Foli and Michele Solimena, showing that beta cells have a huge reservoir of GABA.
Yeah.
And you brought up GAD65. I mean, right. I mean, my first work in his lab was looking at stiff person syndrome, looking at some of the other, frankly, like neuronal proteins like amphiphysin, which are co-released when these cells get destroyed.
And there it is, GAD65, one of the first autoantibodies. So I think that show up in type 1 diabetes patients. This is going to be we are really going to be keeping an eye on this work as it continues. And I'm hoping that this discussion kind of reaches out into the community. And if anyone has any interest in this type of work, they'll reach out to you and communicate, or if they have any interest in collaborative projects that you guys can communicate. And because I do think that coming in from the angle you're coming in, neuroscience, cell biology, and the clinical appreciation of the disorder is a really powerful place to sit. And I think that you could have a lot of great conversations with others in the field. So thank you again, Zach. Really appreciate you stopping by and talking.
Oh, thank you. And again, I'm really just grateful that a forum like this exists. I think it's just such a wonderful thing.
Thank you so much. And I hope you have a great rest of your day.
Thank you. You too.